Okay, so what do we know about what goes through the nucleus? This is an experiment that was done with the T antigen, a large T antigen, the SV40 virus. So when the virus infects cells, it produces the large T antigen. That T antigen is transported into the nucleus of the cell where it shuts down the synthesis of host proteins. And essentially, what we see in this cell here is what they've done is they've taken a large T antigen, right, and they put a fluorescent dye on it. This is black and white, but they've used the green fluorescent dye that we talked about, fluorescein. And so what they've done is they've taken the protein and they've labeled it with these fluorescent dyes. And what you can see is all of this particular protein, this is the large T antigen, is found in the nucleus of the cell. These are all nuclei. What are the little dark dots in here? What are the little dark dots? Those are the nucleoli we talked about, right? Where our ribosomal RNA synthesis is going on. But those are the nuclei, and so if you express it, the large T antigen, the T antigen, or SV40, in a mammalian cell, this is probably a glial cell, this experiment's old, what you find is that they're transported in the nucleus. And the reason they are is because they get transferred, is because they have a sequence that's called the NLS, right? And the NLS stands for nuclear localization sequence. The NLS, if not, it'll be on, it'll be on another slide somewhere. Oops. There's a moment. At least not on this overhead. So it's called the NLS, the nuclear localization sequence. And that sequence is sufficient to move these things into. It was identified by doing site-directed mutagenesis. And essentially what people did is they took cells, these cells, and they mutated those cells at a rate and a level where there would be one mutation per cell. And they then sequenced the proteins, and they looked for proteins that weren't transported. And what they found is that in this region here, this orange lights, you see this region, these charged amino acids following proline. That's sort of what the nuclear localization sequence looks like. They can vary, but they're all preceded by proline and then have these charged lipids like this. If you take one and mutate it, if you take that lysine and mutate it to a threonine, take that gene and transfect it into a cell, let that cell make SV40 or the T antigen from SV40, and then ask where it goes. You can see that now that side of that protein is in the cytosol. It's not transported into the nucleus. Okay. I don't know if I said it the other day when I was talking about signal peptides, I may have not. These sequences are sufficient and necessary to get these things translocated, either transported in the nucleus in this case. But notice it's called transported because they're going through gated channels. It's not translocation. It's moving through a translocon. But in either case, the signals are sufficient and necessary for movement. If you take a secretory protein that has a signal peptide on the amino terminal end, and you cleave it, that protein is no longer secreted. If you take a sequence or a gene sequence that codes for that and attach it to another protein that's not secretory, so the protein is now made with an amino terminal sequence, that protein will be secreted. You can take the gene for actin and with a secretory sequence for one of the immunoglobulins on the end, put it back in the cell, and the actin is secreted from the cell rather than being in the cytosol. So these signals are sufficient. So you know, this is something different about that sequence. You sort of can't tell here, I guess, really, because I said they're usually preceded by proteins. But the nuclear sequences are different because they don't necessarily, they can occur on the amino terminal and the protein, but they can also occur at the carboxy terminal, or they can occur internally to the protein. Right? And most of them are found internal in the protein. What does that mean if they're found internal in the protein? What does that mean about their behavior? Well, when they're on the amino terminal end in a protein that's secreted, what happens? The protein gets attached to the endoplasmic reticulum with its ribosome, translocation begins, and what happens to the signal? It's no longer needed, so it's cleaved, right? But if this guy's in the middle of a particular protein, it is not necessarily on the end. All it has to be is on an exposed region. Will this be exposed on the protein? sure, because it's always charged residue, so it'll be in the aqueous side with hydrogen bonds out to the water, hydrogen bond to the water. So it is exposed. But all it has to be is there, and that gets it into the cell. Does it make sense that it involved that it was inside the cell? Why change the modus operandi? Why not have it on the amino terminal end of the protein? Come up with an idea of why. So what happens to cells when they divide? The cell divides, right? They get ready, they duplicate all of their DNA and all their proteins. The nuclear membrane goes away, right? So that the lost my brain thought went away. So that the spindle can pull the chromosomes apart, right? And go into the daughter cell. Afterwards, the nuclei reform. And when the nuclei reform, they form the membranes from the ER form right around chromosomes very tightly. In some cells, they actually can form around individual chromatin, and they fuse, as they fuse to get bigger and bigger membrane to form a nucleus, actually. But once that nucleus forms around its chromosomes, what's missing from there? Well, what gets out when the membrane goes away? Well, the chromosomes get out, but so do all the proteins, all the nuclear proteins that belong there. All the transcription factors, all the DNA polymerases, and all of that stuff are out in the cytosol. The membrane self-assembles, and what does it do? It reincorporates all of the nuclear proteins, so they're reincorporated back into the nucleus. So every time the cell divides, the proteins get out, and the nuclear sequence, the nuclear localization sequence is reused and given back in. Right? So it makes sense that evolution, rather than having cells make all of these proteins every time they divide, would devise a mechanism whereby they could be reused or reincorporated into the nucleus. And because of that, these guys, it's evolved where these could be internal. So most of the nuclear localization sequences are internal. Some proteins can have more than one nuclear localization sequence. Okay. Do we have a question? That's all we're going to say about that. You'll learn an awful lot more about that when you take some out. So you talked about nuclear localization. We talked about movement through membranes. We talked about vesicles later, I think, if we have time. But I want to talk about what else happens in the endoplasmic reticulum. We started talking about that. Right? We said the proteins that have to be secreted or membranes are transported into the ER. But the ER has lots of other functions. And one of those, which I know I haven't explained, is the proteins that are to be secreted or end up in the membranes are folded in the endoplasmic reticulum. So the folding of proteins occurs in there. And they're very specialized proteins that do that. One of these proteins is called BIP, binding protein. And BIP is in the family of HSP70 proteins. It's like remember there's HSP70 outside in the cytosol, which helps folding. Well, this BIP is HSP70 inside the cell. It's ATP dependent, and it helps to fold protein. It binds to hydrophobic regions while that protein is folding and covers them. But it has another function, and that is that remember this is an ATP binding protein, and it also binds on the protein to be folded. One thing that happens is as proteins are being synthesized and going through the translocon, as they're coming through that translocon, the BIP protein binds to it, it's added to it. The enzymatic activity, or the enzyme itself that adds BIP or helps to add BIP in the membrane protein, and it's very closely associated with the translocon. So as the proteins come through, right, this BIP protein, the HSP70, is added onto the protein. And one of the functions that it plays is that translocon, when the proteins start to go through, they can move in both directions. And in fact, if you have a BIP minus cell, they can sort of stutter. They come in, they go back, and they go back. Once the BIP binds, it sort of blocks that. So it's almost like pulling these through, right? It helps them to go through. I don't know if we put that or not. Why is the reason for that? I didn't say that, but it actually helps to move the protein through the translocon. It doesn't really move it through, but it keeps it going in one direction. It keeps it going unidirectionally through. by essentially binding so it can't pull back out, so it can't stutter. So PDI folds. There are lots of other proteins in there in that endoplasmic reticulum that are important for folding. One of the most important ones is the protein called PDI, protein disulfide isomerase. And protein disulfide isomerase is the protein that deals with what I talked about before, and that's disulfide bonds, right? The formation of disulfide bonds. When disulfide proteins that are disulfide pop through the nucleus. So as proteins are popping through, as they're being synthesized and moving through, if the protein moves through and it has a sulfhydryl group, right? A cysteine residue with a sulfhydryl, and another cysteine with a sulfhydryl, right, is coming in, and as it's folding, those two will form a disulfide bond, right? And it will be between residue 1 and residue 2. If there are then another cysteine, residue 3, residue 4, then it'll form another one with residue 3 and residue 4. So in this reducing environment, that's in that oxidizing environment inside the endoplasmic reticulum, if these sulfhydryls are there, they'll be reduced, right? They'll have these sulfhydryls. They'll be oxidized to form a disulfide bond. Now, in many proteins, the order of the disulfide bond formation is not 1, 2, 3, 4, 5, 6. If you look at the picture, there's one in your book, I'm sure, of insulin. Insulin is synthesized, right? And it's processed, and before it's processed, you get these big things like this, where you have cysteine 1 and cysteine whatever. This is cysteine, and then writes another one here and another one here. So 1, 2, 3, 4. So 1 and 4 might form the bond of a mature molecule, and 2 and 5 might form the bond of a mature molecule. Right? Well, what happens in folding is they're going to be folding like this. So what PDI does is kind of like the PDI has a sulfhydryl of its own, and it's able to reduce those disulfide bonds. Has 2 sulfhydryls, right? And it's able to reduce those disulfide bonds as it becomes oxidized. That's the mechanism, right? It has two sulfhydryls, and they form a disulfide bond, and they reduce these, and they become sulfhydryls. So it opens them up. And what this protein does is it allows the protein to breathe. It interacts with folding proteins, breaks those disulfide bonds after the whole protein is synthesized, and as that protein is breathing, the right disulfide bonds form. So it's another chaperone type of protein that's needed to help these things bind. And there's lots of other proteins. Those are the only 2 that I put up there for you. So folding occurs. The other thing is that this folding is monitored very, very closely. Proteins that go into the endoplasmic reticulum, remember, they're going to be transported to the Golgi. But before they are, cells check very closely, very carefully, to be sure that proteins are folding correctly before they move on, before they move into the Golgi, where they'll then go to lysosome, to the surface of the cell, or whatever. They're processed very, very carefully. Or checked very, very carefully. So in here, there's a quality control mechanism that looks at folding of proteins. And one of the things that's involved in that quality control is this glycosylation we talked about. Remember, as proteins are being synthesized, they have an asparagine level that's in a particular sequence, right? Asparagine, X, threonine. But if those asparagine residues are glycosylated by this core carbohydrate, these 14 sugars that are added by oligosaccharide transferase that they go in there. And these sugar molecules have a lot of functions. We've talked about some of them, right? They protect cells from proteins from being digested by proteases somewhat. They're also serve as ligands for particular receptor molecules. So they have a lot of functions. But the other thing they do is they're involved in this folding process, and I want to try and show you that. So this shows you the quality control mechanism